Hi, my name is Hannah Foreman, and I chose to research the occurrence and removal of pharmaceuticals from water around the world for my global project. As of now, researchers and scientists have identified nearly 100 pharmaceutical compounds as being present in rivers, lakes, streams, and drinking waters all over the world. For the most part, concentrations are showing up in parts per billion and parts per trillion, but their occurrence is gaining them attention from the EPA, World Health Organization, scientists with water research, and the International Journal of Environmental Analytical Chemistry. Most of the studies being conducted are focused on medical facilities because that's where large quantities of unused pharmaceuticals are disposed into areas like sewers. A few examples of drugs found in ground surface waters recently by German scientists are cholesterol regulators, analgesics, and anticonvulsant medications. These contaminants can occur from patients disposing improperly of unused or expired medications, residue left in dispensers and containers, IV bags emptied into the sink, or flushing drugs down the toilet. Lastly, biological excretion should be considered as a way pharmaceutical chemicals can leave a patient's body and enter a waterway. So, a high amount of antibiotics and other medicines are produced for consumption every year, and 30 to 90% of them are excreted without metabolism, meaning our bodies don't break them down far enough. Also, some degradation products are just as harmful as the original medicines themselves. There's a risk for possible development of antibiotic resistance here, which could lead to decreasing effectiveness of antibiotics in the treatment of human diseases. A team from New York recently took on the task of evaluating sewage and waste oil sludge derived materials as an adsorption media to remove the materials used were obtained by pyrolysis of individual waste oil sludge, dewatered sewage sludge, or a 50 to 50 mixture of both at 950 degrees Celsius or 650 degrees Celsius in a nitrogen atmosphere in a fixed bed. Most results of the compounds at environmentally relevant concentrations were completely removed by their efforts of adsorption. It was concluded that factors such as polar surface area, positively charged functional groups, molecular size, and hydrophobicity all played a significant role in whether these medicines absorbed or not. The waste oil sludge and 50 to 50 mixture of both sewage sludge and waste oil sludge absorbed about twice as much material as just the sewage sludge alone. The highest performing adsorbent meant that it had the highest degree of microporosity and smallest pore size. The fact that this process has potential to turn hazardous waste into adsorbents that could be used in waste and drinking water treatment is encouraging news for a sustainable future. Municipal sludge and other industrial waste could be used as a means to clean up something that we have contaminated in our water. In another part of the globe, a team of scientists from Sweden, they've been working on removing the same types of things. They have a concern for the effectiveness of their sewage treatment plants on removing this newly discovered threat. They are concerned about the presence of pharmaceuticals and steroid hormones and the notion that elevated levels of these medications can have an adverse effect on aquatic life. Their samples were obtained from residential sewage pipes, hospital outlets leading to treatment plants, and from different steps within a sewage treatment plant, such as the inlet, the outlet, and between the treatments. The sewage treatment plant used in this experiment had 3 treatment steps: mechanical, which removes coarse particles; biological, which separates and breaks down organic contaminants; and chemical, that removes phosphorus, or phosphorus-containing compounds. A concluding step was employed to pass water through a sand filter before it reached the recipient lake. The highest drug concentrations were not surprisingly collected from hospital sewage streams. Ibuprofen and naproxen with a 4 IBAP was found in highest concentrations. The concentrations detected in this experiment were all in the nanogram per liter range, where it's postulated that resistance by pathogens and other long-term effects can happen. As far as the estrogens were concerned, the removal rates were higher in this Swedish experiment than in Italy and Spain. This was attributed to the tertiary sewage treatment plants employed in these tests. The antidepressants were thought to have absorbed sludge and fat during the primary treatment because their levels were below the detection limit. This team was able to draw some significant conclusions from their research. One of them being that the highest removal rate was observed during biological treatment, which was adequate aside from the drugs diclofenac, estrone, clofibric acid, and oxaflocin. Also, it was determined that a sewage treatment plant with a tertiary treatment is sufficient to remove most of the pharmaceuticals in water, and that traces of these drugs will continue to enter the environment over time. A large part of their conclusions was that understanding removal efficiencies among drugs is linked to chemical structure largely. It seemed that drugs that were persistent owe their strength to chlorine groups also. It was concluded that different treatments, biological, chemical, and physical, were better geared towards removing certain pharmaceuticals. The biological step was especially good at removing some drugs due to sorption to activated sludge and likely not biodegradation. The case was similar during the chemical treatment, where sorption to sludge and particles was most probable for elimination due to very hydrophilic and zwitterionic compounds. Upon analyzing data from removal studies in Spain and Italy and comparing them to the ones found in this study from Sweden, it was concluded that Nordic countries consume a higher amount of pharmaceuticals, which is something I found really interesting. And in yet another part of the globe, scientists from Australia have obtained results from investigating 28 antibiotics in 3 hospital effluents, 5 wastewater treatment plants, drinking water storage catchment, and 6 rivers. Their line of thinking here was that antibiotics in some surface waters have the potential to interrupt important bacterial cycles that are key in nitrification and denitrification, soil fertility, and also animal production. Animal reproduction. This team expressed the main goal of this project as providing baseline information on targeted antibiotics in southeast Queensland surface waters in Australia and regional drinking water supplies. They specified their instrument limit of detection as the concentration equivalent to 3 times the standard deviation of the response equivalent to the noise for the trace of interest. As was expected, 1 or more antibiotics were detected in 90% of the samples. That's a huge amount. Hospitals were a major contributor, but no correlation was found between concentrations and usage volume. It's comforting to know that no antibiotics were detected in drinking water samples. No bacitracin was found, surprisingly to me. Erythromycin was the most frequently detected, and then monosin in the environmental surface waters was there due to livestock medicine as a use. Monosin is also a drug used in beef and dairy industries and has toxicity risks for mammalian cells. During testing, total investigated antibiotic concentrations, or TIACs, were monitored, and it was discovered that the hospital effluent, wastewater treatment plant influent, and wastewater treatment plant effluent locations showed no significant difference in this number. The team recorded a wastewater treatment plant removal efficiency of 80% for the antibiotics studied. Knowing that most antibiotics are only semi-metabolized and rarely excreted in original form, households were another large contributor to these medicines in waste streams. While researchers' removal methods were 80% successful, traces of medicines were still found in effluent samples. This indicates that their processes did not result in complete removal. One difference in the Australian study from other regions of the world was that none of the studied antibiotics were detected in the drinking water itself, while detection of these has happened in other studies conducted in America and Europe. Something that is mentioned frequently in journals is the further analysis of degradation products of these antibiotics. In some cases, these compounds can be more toxic than their parent compounds or pose different threats to the environment. So while this is a fairly new issue, I think the first paper I found was published in 1998, but then most of the research I found was done in 2009 and 2012. Most regulations are being set forth in a qualitative manner by agencies and statutes like the Controlled Substance Act, the Resource Conservation and Recovery Act, or the RCRA, the Centers for Medical and Medicaid Services, and the Health Insurance Portability and Accountability Act. For example, the EPA has recently stated that it is now developing major revisions to the hazardous waste regulations to make them more effective for the healthcare sector and the hazardous waste pharmaceuticals they generate, with the aim of helping states with implementation and the regulated community with compliance with the RCRA regulations for hazardous waste pharmaceuticals. Their website is actually under maintenance. If you go to the PCPP info sheet, while drugs have been detected in most every study conducted and all the ones I researched, the levels are still really low compared to actual therapeutic doses, leading to the suggestion of low risk to human health. Because of this, the development of a formal quantitative value for medications in drinking water hasn't been set by some agencies like the World Health Organization internationally. The stakeholders in this situation are any humans inhabiting regions close to affected rivers, lakes, streams, and drinking water sources, the medical facility officials that are responsible for safety and drug disposals, and investors in the medical facilities. So some of my conclusions are, when looking at the issue our planet continues to face, I think this will only get to be maybe a larger problem if we don't get a handle on it right now. There are multiple things to consider in choosing a removal method. Some methods work best for different geographical areas. Something that works for one region of the world, such as America, might not work best for somewhere in Europe. Things to consider for removal are the chemical structure, the drug class, the molecular weight of the molecule, the surface area, types of treatment steps with a STP, the number of treatment steps, and excretion before or after metabolism of the drug. And so in all my research that I did for this project, I was really impressed with the efforts of different regions of the world to remove pharmaceuticals from water sources. They are proving to be serious about meeting specific regulations that will be set in the future by environmental agencies, and they seem committed to providing safe water. In my opinion, the research groups should concentrate on the degradation products of certain drugs like ibuprofen and naproxen, since those are common drugs in most regions whose degradation compounds have shown to be toxic and present high risks to humans and aquatic life. And so that is my project. Thank you for watching and listening. These are my resources.